Dr. Ramanuj, you are here.
Yeah. Good afternoon. Good afternoon. I am here.
Angaraj sir.
I am here.
Yes, sir. Yeah.
So let everyone switch on the video so that we hear.
Good afternoon, sir. Good afternoon, sir.
Make it live so that we can.
Okay, sir. Live started, sir.
Very good afternoon to the chief guest, Dr. K. Thangaraj, Director CDFD, Hyderabad. Welcome to our Director, Dr. V.M. Tiwari, Director GRI. And we have Dr. Ramanuj Narayan from CSIR-IICT to brief about the Iisf activities. So this is a program under the India International Science Festival, which is an initiative of Ministry of Science and Technology and Ministry of Education with Vignan Bharati, which is a science movement with Swadeshi spirit, led by eminent scientists of the country. The main purpose of Iisf is to celebration of science by all. So the motto is engagement of common people with science in a joyful and entertaining manner is essential for the healthy and prosperous and meaningful life. Through its creative programs and initiatives, Iisf provides opportunities to people and scientific fraternity in the country and abroad to in the country and abroad to come together, work together, and experience the joy of doing science for the well-being of India and humanity. So with this brief, I welcome you all to the curtain raiser and outreach program of Iisf at this. I request Dr. Ramanuj Narayan to explain the Iisf activities in Hyderabad in general and in the country. Over to you, Dr. Raman.
Respected Tiwari sir, respected Thangraj sir, the chief guest of today, my dear colleagues from CSIR NGRI, students who have joined online, and all the viewers of the YouTube online. At the outset, my greetings to CSIR NGRI on behalf of Vigyan Bharati as well as CSIR IICT and India International Science Festival. As rightly explained just before, that how Vigyan Bharati has started this program. This is the 7th edition of India International Science Festival. It was started in 2015 at IIT Delhi. Then second edition was also held in Delhi, then moved to Chennai, then Kolkata. Then last year it was on completely web mode, and this year the India International Science Festival is going to be held in both hybrid as well as in-person mode. So this year, this event, because the festivity of science should be celebrated by the younger generation, the budding scientists, and all the scientists across the country. And this year, Iisf is going to be held on 10 to 13 December at Panji, Goa. And since this event has got more significance this year because it is happening with the Azadi Ka Amrit Mahotsav. And just 2 days back, our Honorable Minister, Dr. Jitendra Singh ji, with Dr. Jain Sahasbuddhe ji and presence of all the secretaries of the different government department, has done a curtain raiser event. This year, this event is coordinated with the line ministry, Ministry of Earth Sciences, and which NGRI is well known in the earth sciences. So this year, that Ministry of Earth Sciences is the nodal agency, supported by all the scientific departments of our country, Government of Goa and Vigyan Bharati. The icing on cake this year is that till last year, DRDO was a regular part, but this year ISRO is also going to be a part of this science festival, and as a nodal institute, NCPR Goa is organizing this event at Goa. This year, the theme of the conference is celebrating creativity in science, technology, and innovation for prosperous India, and the 5 pillars which has been suggested by our Honorable Prime Minister has been put into all the programs. Last year program we are having 41 events, but this year it has been curtailed to 12 events which will be matching with the Azadi Ka Amrit Mahotsav. The 12 programs are Science Film Festival, Science Literature Festival, Science Engineering Student Festival, then Science Village, where I am a part of this, Traditional Craft and Artisan Festival, then Greenwich Book of World Record, Games and Toys, Global India Scientist Conclave, Eco Fest, and New Age Technology Conclave, and then there will be a very big expo, that scientific expo will be organized. So I request that let us join, because today CSIR NGRI is doing the curtain raiser as well as outreach program, that today whoever is linked to us, it's our 10 people. If we are reaching to 10 people more, so at least whoever has joined today, it will reach. So the main motive that our scientific message should go to the each and every person. So please join. Lot of program will be online. If you are not able to go to Goa due to COVID restrictions, so you can see the online 700 film will be telecast. All the scientific demonstration will be done. So my urge that all of you should be a part of this festival by virtually, or if you join with personally at Goa. So most welcome on behalf of Vigyan Bharati, Iisf organizing committee, and on behalf of Hyderabad, Telangana, Vigyan Bharati, I do invite all of you to be a part of this function. Thank you, sir. Thank you, Tiwari sir, for giving this opportunity. Thank you.
Yeah, request our Honorable Director to address and introduce the speaker, address the gathering as well as introducing the speaker. Over to you, sir.
Thank you, Dr. Sarma. Today, distinguished guest with us, Dr. K. Dhanraj, guest in a home. So first, I take opportunity to welcome the today's chief guest, Dr. Dhanraj, to this outreach event of CSIR NGRI, and all those who had joined through online media in this important event of bringing together creative minds, which is being celebrated as a part of our festival at NCPUR Goa under the auspices of Ministry of Earth Sciences. I also welcome the Vigyan Bharati representative and senior scientist from CSIR Indian Institute of Chemical Technology, Dr. Ramanuj Narayan, and several of our young creative minds, those who have joined to YouTube. You must be wondering what does it mean to have an outreach of a science. And You may also be wondering, why do we need to have it at all? And one of the basic requirements for the society to grow and engage with or confront with the things which are coming in Anthropocene. Sometimes they are man-made and sometimes they are driven by the variability in the system that could be natural as well. And having this opportunity of outreach, we cannot have a better person who have used the modern technology to trace the human migration and to understand the genome of human for different purposes. And this will be much more exciting. This had, as Ramanujji has said, that icing on the cake. So it's a icing on the cake of the outreach program in the Hyderabad, having our distinguished guest today is speaking to the large number of audience connected through online. We all know that the whole country is celebrating Azadi Ka Amrit Mahotsav, and we want to understand, we want to know, and we also want to make our young generation know that how the freedom struggle was fought. What has been achieved? How do we, in 75 years, grew up with the ideas of independent thinking, and what action have been taken to make our country progress and march with the advanced countries in the world, and take the resolutions that how will we take our country forward when we celebrate the 75 years of India's independence. And in all that, you must be wondering that why do we need to have science at all, reaching to the common people? And if we see the really the circle of the growth, development, and sustainability, the humans are in the periphery and the center is for entire progression is science and technological and innovations happened during the last century. And this is what we want to catch up. This is what we wish to do together and inculcate this culture in our young mind, our ignited mind to take the country forward for the betterment of the people in the country and in the globe. In this respect, there are several programs being done at CSIR-NGRI, and during these 75 years of the celebrations of India's independence, CSIR is celebrating the 80 years of its important contribution to the society of the country. We also at CSIR-NGRI have been celebrating our 60 years of scientific excellence and services for the mankind. In this respect, the CSIR's contribution, whether we start in the making of the the indelible ink, or whether we had to have first tractor in the country, or talking especially to the contribution of our own institute having the first instrument flying on the Indian soil for the geophysical exploration, are making several new technologies developed, adopted, innovated for economic and the societal applications. We have today, as I said, that none other than one of the very distinguished scientists whose work directly connects to society, and most important is one of the very good human beings we have ever encountered in our life. Dr. Csir Contribution, the director of the Center for DNA Fingerprinting and Diagnostics at Hyderabad. He is also since very long time been associated with CSIR as a researcher in the different capacity at CSIR Center for Cellular Microbiology, Hyderabad. You all know, or probably those who are young kids, the school children, for them, I would like to say few things that his main interest, scientific interest, is the origin of the modern human. You know, which will be so exciting to know how the human being evolved over thousands of years, and how did they migrate as well as working on cardiovascular diseases and many other important biological scientific issues and male infertility. He is known for his publications in the leading journals, which appeared in the Science and Nature kind of the journal's front page, and authored more than 270 research papers in the leading journals. He is elected fellow of two distinguished science academies of our country, Indian National Science Academy and Indian Academy of Sciences, India. Presently, he is also a J.C. Bose Fellow and been awarded with several awards, and to name few, Sun Pharma Research Award in the Medical Sciences, Raman Research Fellowship, Lifetime Achievement Award, in Excellence and Science Award, Distinguished Scientist Award, Dr. Subhash Mukherjee Oration Award, and so on. I can read a very long list of the awards and honors, and you can learn all of them from his website. I will not take long to introduce our well-known guest today, but I would like to emphasize to all of you that how important it is to take the science for the purpose of betterment of the human being can be learned from the work from Dr. Csir Contribution. I also take this opportunity because we are organizing. Yes. We are organizing this event as a part of outreach of India International Science Festival, and as Dr. Ramanuj Narayan had explained to all of you in great detail about the events, I once again urge all of you to log on, get connected with this very inspiring event plan for celebrating science and making people excited to be part of the science movement. That does not mean only doing science, but also promoting scientific culture, also inculcating the science in the society, at home, in office, and following all standards of the modern age science. With this, I once again thank our guest today for being very kind to agree at a last moment request to deliver the lecture for the students, and also to the Viva representative, Dr. Ramanuj Narayan, to be with us when we get connected to the large number of students from the different schools and also the researchers from the different domains of science. Thank you very much. Dr. Sharma, it's over to you.
Yes, sir. Thank you, sir. I request now Dr. Thangaraj to take over. Over to you, sir.
Yeah. Thank you. Good afternoon to all of you. Thank you, Dr. Tiwari, for your kind invitation and the kind words. Good afternoon to everybody. It's indeed a great pleasure for me to talk to this event, India International Science Festival event. Earlier, I was part of organizing some of the events, and also I was giving talk in some of the events. So it's a pleasure again to talk in this. And thanks, Dr. Ramanuj, for introducing the Iisf, and also thank Dr. Srinivas for organizing this event on behalf of NGRI. I also welcome all the colleagues from NGRI and CSR family and all the participants, particularly students. I hope that my talk would be very useful for all of you. So with that, I would now start sharing my slide. My slide is visible?
Yes, sir.
On full screen mode?
Yes, sir.
Okay, great.
Please go ahead.
Okay. It's a topic of interest of everybody, how our own species have originated, right? And also you may be wondering that what is the connection between our origin and our health and disease? So I want to just quote what the well-known evolutionary biologist Dybzhansky has given a title in one of his articles in the American Biology Teacher. He said nothing in biology makes sense except in the light of evolution. So it means that so whatever the science we do, the biology particularly, that if you don't understand the evolution, you will be missing something. So towards the end of my talk, you will be appreciating how important this statement is. You all must have read about Charles Darwin and his work, where he has written about origin of large number of species in his book called The Origin of Species, right from Archy to whales. But what was missing in this book was how human might have originated. There was one more evolutionary biologist, Thomas Henry Huxley. He wrote a book called The Evidence as to Man's Place in Nature, where he was arguing that we all must have descended from apes. Then after 8 years, Charles Darwin wrote another book called The Descent of Man, where he declared that chimpanzee and gorilla are our closest living relatives based on the anatomical similarities we have. One would wonder that how we all evolved and what is the statement was made by both Darwin and Huxley is that we all must have descended from apes and chimpanzee. And now it is well known that human and chimpanzee must have diverged from a common ancestor called hominids about 6 to 7 million years. It's not that suddenly the ape or chimpanzee turn into human. But again, before we are what we look like today, there were several species have appeared and all vanished. During the last 6 to 7 million years, there are a large number of species have evolved and also they have disappeared. In this process, there are several anatomical features have changed. There are several features including the ability to walk with the 2 legs, the ability to make tools for killing animal or hunting food. And this brain size has increased tremendously. If you look at in the early species, chimpanzees, the brain size was as similar as chimpanzee. Then it has increased to 35% of the modern human in case of Australopithecus. In Homo habilis, increased to 50%, Homo ergaster 60%. Then suddenly in Homo neanderthals, our closest relative in terms of evolution, where people have predicted that Neanderthal had slightly larger brain size compared to human. But this species no more, but nobody knows whether this species was intelligent or not. But interestingly, there was one theory proposed by Paul Mullers from Cambridge that inherent violent behavior of modern human probably responsible for vanishing of this species. I don't know whether it's true or not. Then if you look at scientifically that why all these changes, anatomical features and many other changes happen during the course of evolution. If we and chimpanzee are 98% of our genome are similar, why we are looking so different? And this 98% of the genome are not continuously exactly the same as what chimpanzee has. But there are a lot of rearrangement of genome or shuffling of the genes and the chromosomal segment. Just giving one example, taking just one chromosome, that's a Y chromosome of human and other non-human primates. So one of the genes called SRY, that's a sex determining on the region on the Y chromosome that is essential for making individual as male. That is on the short arm or P arm of the human chromosome, but same gene is present in the long arm of chimpanzee and few more species. But again, in the orangutan, it is on the short arm. If you look at few more genes, like chromodomain Y, deleted azoospermia, they're all in the long arm of the human chromosome, but short arm of the chimpanzee. And again, in this case, orangutan, there's one copy which is also present in the short arm. So what all this tells is that during the course of evolution, there's a lot of DNA rearrangement, DNA modification, changes, mutations, a chromosomal segment itself either deleted, duplicated. There are many more things. So the evolution is actually parallelly goes with the genetic variation, genetic mutation. So whatever the change we are seeing now is all because of the changes in our DNA or genome. So the question after having all these is that how did unique human feature originated? How was the capacity for language, reason, and culture encoded in a gene during the course of evolution has taken place? As I mentioned, that there is a genetics, as a geneticist, I strongly believe that the genetics is behind everything. Like, for example, we talked about brain size. There's a gene called myosin heavy chain. And that gene, the variation of that gene, when you compare to chimpanzee and human, is different. Because of that change, there's a more space created in the brain. Therefore, the brain size has increased. Similarly, there are few more genes. Another gene which is important, which involved in learning, the experience of pain, perception, behavior, and memory, is the protonorepinephrine. FOXP2 is the gene which is essential for normal human speech and language. That means, so whatever the DNA change happened during the course of evolution is responsible for the today how we look like. If you still don't believe, then you look at, there are three brothers who born to a normal parents, but they're typically having a feature like chimpanzee. And what would have happened here, that whatever the mutations happened during the course of evolution, probably there's a master regulator gene where there's a mutation, back mutation, which brought feature like chimpanzee. So it's a clear evidence that genetics is behind everything. This is another example where you see it's a body full of hair in this child. And this is one of the features we have lost, where you see chimpanzee has body full of hair, but we have lost during the course of evolution. But again, what you see here, there could be a back mutation brought the hair on the body. In addition to whatever the genetic change happened, this is one of the interesting paper where the authors believe that there was a self-domestication. We know that the cattle or dog or something which is in the wild, we bring home and we grow them at home, then it become domesticated and become very soft. Right? Similarly, no one has domesticated us, but human themselves has domesticated, and we become like a cooperative species. With this background, let's see how and when the modern human originated. It is now well established that the modern human in the sense that the way we look like today, originated in Africa about 160,000 years back. And there are several scientific fields have been put a lot of effort in understanding about how the modern human migrated out of Africa, including geneticists, linguists, historians, archaeologists, anthropologists. And would be surprised to know that even geologists in this paper, earth science and geologists, so they have drilled East Africa, and they found that there was a lake, Malawi, and there was a mega drought during 135 to 75,000 years ago. Therefore, no water, no food. Therefore, people started migrating out of Africa. And another question is, from Africa, how did they migrate and populated the rest of the world? There are two hypotheses. One group of researchers includes geneticists, archaeologists, anthropologists, linguists, historians, and many more fields. They believe that the modern human must have started migrating from Africa towards north. It's called as northern land route. But other groups did not believe on that. They thought that the climatic condition might not have favored those time to migrate towards north. They must have made attempt, but they must have come back. So they believe that the modern human might have migrated taking the southern coastal route. And mostly they're all based on the archaeological evidence present in some of these places, including Australia. It was predicted that the modern human might have arrived Australian continent around 50,000 years back. China 39,000 years back, Middle East 47,000 years back, and Europe 40,000 years back. Unfortunately, we don't have any archaeological evidence to suggest that when did the modern human arrived in Indian subcontinent because of the climatic condition. Although there are some place where there was excavation took place. This is a place called Jivalapuram in Andhra Pradesh. There was excavation in the year 2007. They found a lot of tools, stone tools, which the modern human might have used, but unfortunately they did not find any biological material of a modern human, including bone, hair, nothing was found. There is one very strong evidence recently found. There was an intact skeleton of a woman in Arapan region, particularly a place called Rakigiri, where we analyzed the genome, but unfortunately it's not very old genome. This was about 3,000 to 4,000 BCE old. And it has a combination of the ancient Indian genome and also the Iranian hunter-gatherers, not the Iranian farmers. But again, it's not saying that when did the modern human arrive in the Indian subcontinent. There are few more evidence, particularly in deep south near Madurai, one place called as Kiradi, other one is Adityanallur. Although we are analyzing the sample, but I don't think that's going to give any view about the early human migration. But in the absence of any of the archaeological evidence, we thought that analyzing the Indian genome would answer important question. You can see India is very unique country where we have very huge amount of diversity. We have a group of people who typically look like Africans with very dark skin color. We have group whose skin color is very light, typically look like the Europeans. Some group look like Southeast Asian, and some group is very unique to India. They doesn't look like anybody else. So with all this, we have now in India 4,635 anthropologically well-defined population group in India. That includes 532 tribes, 72 primitive tribes. That includes 36 hunter and gatherer lifestyle. That means they are just hunt for their food. They don't do any job, including agriculture. We have been working on this field for last nearly 2.5 decades and analyzed several hundreds of populations, thousands of samples. While doing, we are surprised to know the first out of Africa human is in Indian subcontinent. Who are they? They are none other than the tribal populations of Andaman and Nicobar Islands. How many of you are aware that people with typical features of Africans are in Indian subcontinent? They are in the islands of Andaman and Nicobar. There are 572 islands, which is about 2,000 kilometers from Chennai and 2,000 kilometers from Calcutta. There are 4 tribal groups who typically look like Africans: Jarwa, Great Andamanese, Onge, and Sentinelese. these three tribes have some contact with the people who migrated from mainland, who settled for a very long time. But the Sentinels are the one who still in isolation. Nobody can go near them. They will shoot with arrow. That happened about 2 to 3 years back. There was a American tourist tried to go near them, and they have killed him with arrow. So what we did, we have collected the sample from the three other tribes, Great Andamanis, Onge, and Jarva, and we used different kind of genome. Initially, we used the mitochondrial genome. For the students, in a cell, will have nuclear genome. In a somatic cell, there will be 23 pairs or 46 chromosomes. Of that, 23 we inherit from the father, 23 from the mother. In addition to that, in the cytoplasm, we'll have large number of mitochondria. Or you all know that mitochondria is the powerhouse of the cell. And in each mitochondria, there will be large number of mitochondrial DNA, which is circular. And if you take a cell, you might find hundreds of mitochondrial DNA. And the mitochondria number is not fixed. It varies from cell type to cell type. Wherever there's more energy requirement, the number of mitochondria will be more. Same way, the mitochondrial DNA copy number also will be very high. While we inherit the nuclear genome equally from both the parents, 23 from father, 23 from the mother, we inherit the mitochondria and mitochondrial DNA only from the mother. Because the mitochondria in the sperm will be in the midpiece. At the time of fertilization, the midpiece and the tail remain outside. Therefore, there's no contribution of paternal mitochondria. So we sequence the mitochondrial DNA of the Onge, Jarvas, and the Great Andamanis. And these are just to show that there are two sequence, a short sequence of mitochondrial DNA. But you can see here is a difference. You know that DNA is made up of 4 nucleotides, adenine, guanine, thymine, and cytosine. They're all differently labeled here. And you can see there's a difference at this position. There's a G, whereas it's A in the other sequence. There's an A here, G, A, G. So these are the changes you see generally in the mitochondrial DNA. But we take the changes which are highly conserved and try to construct the haplogroup. And the set of mutations makes a specific haplogroup. That means each haplogroup, these are the haplogroups, and these are defined by a set of mutations. And you can clearly see that the people in the Africa have L1, L2, and L3 haplogroups. Of course, those who have migrated from Africa, like Siddhis, some of you may aware, the Siddhis are in Gujarat, Karnataka, and also in Hyderabad. We also found some of them carrying this L-derived haplogroup. And after this, the additional set of mutation, they will fall into M, R, N, and again, every haplogroup will have defined set of mutations. This is to show that how the worldwide populations are with different haplogroups. When we sequence the mitochondrial DNA of the Andamanis, we found there is a large number of mutations which are uniquely present in the Andaman tribes, but not in any part of the world. The number indicates this is a position in the DNA sequence, there is a mutation. 200th position, 1524th position. So these are uniquely present there. While seeing the phenotype, we expect that they would fall into L1, L2, or L3 haplogroup, but no, they have unique mutations because they are living in the island for last several thousands of years. Therefore, they acquired unique set of mutations. One can also calculate the age of the population based on the mutations. Okay. So based on that, we concluded that Andaman tribes are the first modern human who migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. So this is the first genetic evidence suggesting that Andamanis are the first modern human who migrated out of Africa and settled in Andaman and Nicobar Islands about 65,000 years back. Subsequently, there are many studies, but I'll not go into detail. So now I come to the nuclear genome. So after that, there are several studies, and then after human genome was sequenced, the variations in the genome across the world have been identified, and now there's a 1 million genetic mutation, particularly SNPs, are placed in one small chip, and that can be used for evolutionary study. So we use the autosomal markers, which are contributed by both father and mother, but the number is 1 million, very, very high number. So therefore it would be much more useful, not only for understanding the evolutionary aspect and also for some of the medical aspects. So we used the 1 million SNPs. Now we carefully selected populations across India, across the social groups, so-called upper caste, middle caste, lower caste, tribes and primitive tribes, and also all the linguistic groups, including Dravidians, Indo-European, Austroasiatic, Tibeto-Burman, and Andaman and Nicobar Islanders. So then we also compared sequence of the Human Genome Diversity Panel, and HapMap is this marker, the consortium where they have sequenced and identified the mutation from different countries. And we compared those sequences, and we came to a conclusion about India, what happened in the prehistoric India, and how the Indian population structures. So what we have done was we used all the Indian populations which we have studied, including European, African, and Chinese HapMap dataset. So this slide, this is the PCA plot, gives you approximately some clue how complex the Indian populations are. While you see that European, there are large number of samples, but they're all in a single cluster. Similarly, Africans and Chinese, suggesting that genetically they are more homogeneous, not much different. Therefore, they are in a single cluster. Whereas look at Indians, every single population is unique. Although we all unique, but still some kind of genetic affinities between the groups are existing. Otherwise, they would not have formed the gradient of cluster. They would have cluster somewhere here and there. There's a difference, but still we have some kind of affinities. Then what we proposed was that the Africans and Andamanis divergence is deep-rooted because the Andamanis migrated from Africa very long back. While migrating through southern coastal routes, some group has stayed back in southern part of India, whom we call as ancestral South India. Then there was a second wave of migration from Africa towards north. One group has gone to Europe, other group has come to northern part of India, whom we call as ancestral North Indians. These are the two founding populations of India, ancestral South Indian and ancestral North Indians. And these two populations have admixed and gave rise to many population groups. To understand that when this admixture took place, this admixture between ancestral North Indian and ancestral South Indians, so therefore we studied large number of groups from South and North based on the recombination. Every generation to the meiosis, there will be a recombination between the homologous chromosomes. In the somatic cell, I said 46 chromosome, but in the germ cells, like egg and sperm, there will be only 23 chromosomes. So before that cell division, meiotic cell division, there will be a recombination between the homologous chromosome, the pair of the chromosome. So taking that clue from that, we estimated that initially the ASI and ANI gave rise to many populations independently. Then during the last 2,000 to 4,000 years, these two groups, ancestral South Indian and ancestral North Indians, have admixed and gave rise to many population afterwards. Then the admixture stopped about 1900 years ago. Then every single population started maintaining the endogamy, means marrying within the group. So initially 2 founders gave rise to many populations, then there was admixture, then the endogamy marriage practice. That means marrying within the group. So this was much before the caste system. The caste system was much later. Probably the caste system must have put more pressure on this population to maintain the endogamy. So now let's see what is the impact of this endogamy, marrying within the group. So to understand that, now we took large number of populations, 275 population, and we took 2,800 individuals from this, and you can see distribution of that. This time we did not restrict only within India, but we have gone to the entire South Asian countries, including Pakistan, Sri Lanka, Nepal, and other countries. And where we found something interesting, that most of the Indians or South Asian groups are likely to have a population specific recessive mutation which might cause the population specific recessive diseases. And this tells you that how we predict that the most of the population in India are having mutation which is responsible for population specific disease. This is called as IBD score, identity by descent. Each line represents one population. Then there are 2 red color ones, which are Ashkenazi, Jewish, and Finnish population. These 2 populations are well studied, and they practice endogamy like the Indian population. But still they are with very tiny IBD score. The more length of the IBD score, that means more chance of having a population specific recessive disease. That means we don't have a clue of most of the populations which might have a population specific disease. And these are some of the examples what you see, the Ashkenazi Jewish large number of diseases, and these are the genes, and most of them are autosomal recessive. I'll tell you what is autosomal recessive and autosomal dominant. So I mentioned that we have pair of chromosomes. If you take chromosome 1, for example, one chromosome carries the mutation, other one is normal. If the person carrying the mutation, one of the chromosome in heterozygous condition, have the disease called as it is dominant, because one chromosome, despite its normal, there's expression of disease. So that's a dominant. But in the recessive, mutation in one of the chromosome and other chromosome is normal, the individual will be absolutely fine. There will not be any phenotypic effect at all. But what happened when the population practice endogamy, marrying within the group, the mutation which causes the recessive disease will increase in the population. But after several generations, then there's a chance that 2 individuals who are absolutely fine, but the carrying a mutation in heterozygous condition, that means only one chromosome carry the mutation, other chromosome normal in both father and mother. But when they marry, there's a chance that they contribute the abnormal chromosome to the both of them contribute the abnormal chromosome to the next generation. Therefore, there's a disease. To explain that, you see that this is a heterozygous recessive mutation. Therefore, there's no disease in this individual. This is called as pedigree. The circle indicates female, square indicates male. If you put line, there's a marriage between these 2. And if you put a line down, and these 2 are their offspring. You can add, if you put again, extend this line and put square, that means there's a son, these 2 are daughters. Okay. Now, you imagine that I was mentioning the pair of chromosome, one chromosome carry the mutation. If it is a recessive, there will not be any problem at all. If it is a dominant, then there's a disease. So consider this is a recessive disease, the recessive mutation. then this mutation goes to next generation, both the daughters. Both of them are marrying somebody outside without this mutation, therefore no problem. It's again going to the next generation. Going to the next generation, again here. Similarly here, this daughter transmit to this daughter, her daughter, then she transmit to her son, then goes to the next generation. Now, after several generations, they might have lost their relationship. Now they are unrelated, but both of them are carrying the same mutation. But imagine that if they marry, there's a chance that 25% of the chance that they contribute these two chromosome, abnormal chromosome, therefore there's a disease. So that is very, very common among the Indian population. To explain that, you can see this is one disease, polymerase gamma is the gene. If that has a mutation, that will lead to multiple mitochondrial DNA deletions. This is intact and this is a deletion. And this is a wild type with C. This is a mutant T, C2T mutation. This is heterozygous, where you see both C and T. And this family, you can see that the father carry the mutation in one of the chromosome, mother also carry the mutation, but they are absolutely fine. But unfortunately, they transmitted these abnormal chromosome to two of their children, and one son died at the age of 13. We don't know the fate of the child. Unfortunately, one son has heterozygous, and this would help them in actually tracking this mutation. And if they grow and ready to marry, they can actually test for this particular mutation in the girl whom he is going to marry. Therefore, they can actually prevent such kind of disease in the future. So with having all this, what we need to do is something like what the Ashkenazi Jewish practice. So they practice, they have an organization called Doreshim. It's a community genetic testing organization. They go to school, they collect the sample from the children, sequence and put it in their database. And they grow old and they are ready to marry, both boy and girl. They go to the organization take out the sequence and compare and see whether both of them are carrying some of the well-known mutation which they characterize. If both are carrying the mutation, then the advice, give the counseling either not to marry or even the marry, they should look for this mutation, the fetus, whether it's carrying the mutation homozygous condition or not. So that way, they have been practicing this for last several decades, and they have eliminated a couple of diseases by doing this systematically. So in this direction, we are now looking at some of the population where we found high IBD score. We also looking at some of the population, some of the disease which are restricted to some population, like Handicap disease. There's a place called Karnataka where the people are, most of them are with short stature, with bone abnormalities, and they get huge amount of pain. So we are sequencing such population. This is another one, Udanam kidney disease. There are a large number of such diseases are being now sequenced, and hopefully we'll be able to identify the mutation responsible for this disease. Then we screen the population, give proper counseling. Therefore, we can actually avoid such kind of disease spreading into the next generation. So that's the future work. So with that, I would like to thank all my colleagues, including Dr. Lalji Singh and many of my colleagues and students. There are a large number of collaborators involved in this study, and funding has come from CSIR, UKRI, DBT, DS, and the Medical Research Council. Thank you very much.
Thank you, sir. Excellent presentation. Director, sir, over to you.
Thank you very much. I think we will not have any questions. And if there are any queries, please put in the chat box. We'll collate all the queries and request Dr. Kandaraj to reply them, and again we'll put them. So let's we'll move to the closing of our outreach program today. And Dr. Sharma, you may invite Dr.
Dr. Tiwari, sorry, can I leave because I have to attend my student colloquium.
Yes, please.
Right.
Thank you very
Sorry,
much.
leaving in between.
Thank you, sir.
Thank you once again for inviting.
I request Dr. Nimisha Vedanti to propose formal vote of thanks.
Yeah. Now that our chief guest and the speaker has left, so maybe he can see this recording later, that on behalf of our institute, we were really very thankful for his wonderful talk, where he really took us from human evolution to very interesting topics like back mutations, migrations, then the history of Indian population, and what are the cures we can have for the genetically related diseases. So in fact, I wanted to thank him on the behalf of audience also for his for the time he could really spare from his extremely busy schedule. But that's really, we were really lucky to have this talk under this Iisf umbrella. Now, I also like to thank Dr. Ramanuj Narayanan for gracing this occasion and sharing the information on the upcoming India International Science Festival happening in Goa in December 2021. And our director also, who explained us that what is the importance of such outreach programs. And I sincerely hope that with these talks and our director's address and Dr. Ramanuj's information on Iisf, many people would get interested to attend and participate actively in the Iisf program. And since this program was held virtually, it's my duty to officially thank the IT group and the information group, and also the organizing committee of this program who successfully conducted this program. And congratulations for such a wonderful talk, especially Dr. Srinivas Sharma. Thank you so much.
Yeah, thank you, everyone, for joining on MS Teams as well as on YouTube.
ठीक है शर्मा जी, बाय बाय.
Thank you, Narayan.
शर्मा जी डायरेक्टर कॉन्फ्रेंस करने का है, ठीक है. कॉन्फ्रेंस है.